This is only a test.
Hello, and welcome to another episode of This is Only a Test. I'm Mickey Urdea from Halteris Associates. And today I'm going to talk about aptamers. And I'm just going to tell you up front, I actually think this is the most underappreciated technology in all of immunodiagnostics. And I'll explain why I believe that. So we're still in a great need of better immunoassays throughout industry, research, academia. And there have been remarkable improvements in some areas. For instance, LC mass spec, the company C2N, for instance, which has a series of neurology immunoassays, for instance, the different tau proteins. I mean, they're at the picogram per mil level. But, it doesn't require antibodies, which is wonderful, but it still takes expensive equipment and highly trained people to run it. We've got digital immunoassay technologies from Quanterix and previously from Singulex, and they've improved the limit of quantitation dramatically, but they're still limited by the availability of those instruments and that technology itself. In fact, Singulex went out of business, and they're also limited by the size of a multiplex that they can run. How many immunoassays can they run simultaneously in one run? So, and one of the major reasons for that is, and this is the fundamental issue that I'm going to talk about today, is that it's the quality of the reagents, which is one of the major problems with these tests today. It's the quality of the reagents. It's not the systems. It's not the signal they can generate. It's the quality of the reagents, and I'll explain why. Today, these reagents are almost all monoclonal antibodies, and they've worked really well, and I'm going to talk about some of the improvements that are occurring in that whole area. But I'm going to focus today specifically on aptamers. I'm going to talk about why I think they're underappreciated, the major problem of immunoassay being signal to noise, not signal, and the advantages of aptamers, as well as exactly what they are, how they're made and selected, which is really key to their attributes, and some of the major types that are now available. Also talk a bit about some of the shortcomings of aptamer technology. Finally, I'm going to describe why they aren't universally used in immunoassays and why I think they should be. So we've known for a long time that there are enormous varieties of proteins in typical samples, like say a blood sample, and the range in concentrations is very large. There's a famous paper from way back in 2002, Anderson and Anderson, has been reported many, many times, and this figure is from that. And they point out the fact that there is about a 10 order of magnitude difference in the highest concentration and the lowest concentration proteins that people are looking at. However, that was based on the limited detection of the assays at the time. And what I'm showing this little red box here is that there are a lot of things at very low concentrations for which at the time there were very poor immunoassays. Even today, we really struggle with, for instance, some of the interleukin assays that are at such low concentrations. And I believe that's because of the reagents. So let me start with describing the most popular format of immunoassay for many different types of systems, this being both in research and in vitro diagnostics. And that's the classic sandwich immunoassay. We've typically got in a sandwich assay one antibody, shown at the bottom left there, which is bound to some sort of solid phase. Doesn't have to be in that format, but usually is it. That could be a microplate, like an ELISA plate, or it could be, for instance, a bead. We then have shown here a little square, an antigen, a protein antigen, usually, that we're looking to detect. And then we have a second antibody that is labeled. Could be labeled with a fluorescent label, could be labeled with chemiluminescent label, could be an enzyme label. Lots of things that are used for that. Lots of tricks in the trade. But at the end of the day, what you're trying to do is to form a sandwich where that antigen is creating the sandwich by being the meat, if you will, between the two slices of bread, with the label being bound because the antibody that the label is bound to is bound in turn to the antigen, which in turn is bound to the binder, the capture antibody. I'm going to use that term here in terms of capture reagents. Now, what I'd like to talk a little bit about is how this works, and then where the problems are. So For some of you, this may be extremely boring, and you might want to skip ahead a little bit, but let me try to explain what we hope is going to happen in an immunoassay that's done in this way versus what actually does happen. So this first figure here is more or less a very clean assay situation. So I'm showing over on the left here in the first panel that uh, there's one sample, let's say this is a control sample that has none of the antigen that we're looking for. Again, we're looking at that square, the purple square. Um, and so that's on the left-hand side, and then on the right-hand side is the sample that has the antigen in it. And right now I'm showing it as basically buffer with and without antigen, the best possible condition to run an assay like this. So the way this is done is first there is a binding step which occurs in that first phase where we're permitting the antigen in the sample to bind to the capture antibody on the surface of a well here in this case. And then the next step is to allow it to bind for some period of time, wash it, and then in panel 2, add the labeled antibody. So that is added, and we incubate again, make sure that that has an opportunity to bind to the antigen that is bound to the capture antibody. And if that's the case, then we're going to get the label bound. So on the left-hand side, we don't see any binding because there's no antigen, and on the right-hand side, we see efficient binding. And then finally, in 3, after we wash away everything that's not bound, we're looking at the detection on the left in the control. We should get very little signal there. And then on the right, we should see a significant amount of signal, and we're looking at a signal in a noise level of 0.3, I'm making this up, by the way, versus, let's say, a signal of 3. So that gives us a signal to noise of 10 on the right-hand side. That's all in theory. But now let's go to, the reality here. And of course, the reality is in most cases, what we'll typically have in a negative sample is a lot of other material. Let's say a blood sample with a lot of other proteins in that very large dynamic range. And then we'll have in a situation where perhaps it's an infectious disease. You'll see the antigen that's present in the sample versus the control where there should be none of it. And of course, in most cases, in looking at host antigens, we're looking at different concentrations of them. So perhaps on the left-hand side would be a very low concentration, on the right-hand side, a very high concentration. So same sort of thing. We permit the antigens to bind, but we're going to get some of those other materials binding as well in more than one way. They can bind to the capture antibody, which is very common, and sometimes they'll bind to the surface of the plate. So we try to prevent that with particular materials to decrease that binding, but it still will occur. And so what we've got on the left-hand side now in the middle, number 2, is seeing those undesired, unintentional sandwiches forming. And then on the right-hand side, which is much more complicated, it's got all that junk plus the things that you really want. So now you've got a combination of specific and non-specific binding that's occurred that leads to label being bound to the plate in a control and in the actual sample. So then on the full right-hand side, number 3, we're looking at the left-hand side where all those non-specific things are still bound to some extent, and then hopefully we get a higher signal in the case of the actual sample, which gives us then on the bottom there the noise, which is higher than the last one I showed, of course, because now we've got those other things to look at that are non-specific, and we still have the specific signal, which was 3 on the previous slide. It's 6 here because it's the combination of the noise and the specific signal. So that gives us a signal to noise of 2. So here's our problem. The problem is that non-specific binding which occurs that leads to the noise. And that's why I'm saying I do believe in most cases, in most immunoassay systems, we've got enough signal. We just have too much noise. We got to get rid of that. There are a lot of ways people are looking at this. And I'm going to focus specifically on binders, but just to tell you, there are differences in configurations. I think I talked before in the proteomics episode about Alamar and their method for immunoassays. Very clever way of getting rid of non-specific binding. But I'm going to talk here about the use of reagents to do that, so that this is applicable to any immunoassay system. So our traditional binders, as I mentioned, are antibodies. We started out with polyclonals, moved to monoclonals. They've been extraordinarily valuable, but we're seeing a lot of innovation there in actually engineering the antibodies themselves. So this is being done in a number of different ways, but we're seeing improvement in standard monoclonal antibodies by actually engineering them for various applications. So that's one way, but there's still a lot of non-specific binding. Looking at different types of naturally occurring antibodies, camelid antibodies from llama, camels, etc. Strange little things. They're smaller. They bind very tightly. They stand up to heat. They've made some inroads, but going further than that, there are things such as nanobodies, many of which have been created from camelid, but they're very small, try to decrease the amount of non-specific binding. Then one of our favorites is antibodies, which was also developed by Alamar. Those antibodies have proved very useful in a number of different ways. They're binding to two different epitopes, and that increases both specificity and sensitivity. But now I want to switch to aptamers and talk about some of the unique aspects of those. On the far right there, I showed something called molecular imprint polymers. Very interesting technology, been around since the mid-90s. I remember the first paper I saw on this in, I believe, in 95 from Klaus Mosbach at Lund University. Really interesting technology for a number of, I think, chemical reasons. It's been difficult to apply broadly. But aptamers is what I want to focus on now because there's been a lot happening over the course of the last many years that I think makes them even more attractive than they used to be. Okay, so I'm going to state up front that I have a bias towards these aptamers for reasons I'll discuss. I think most people in this field will be able to say that the following are particularly desirable attributes of these things. One is better assay performance, better limit of detection, better limit of quantitation, dynamic range improvements, CVs, analytical specificity, all these things. I mean, I'll come back to this, as being improvements that can be made by using aptamers instead of antibodies. They're robust molecules. They stand up to heat. They're biological degradation. There are ways in which you can protect it from nucleases. Can vary pH, can vary ionic strength. And there are people that are actually developing assays in things like organic solvents, believe it or not. So there are a lot of things you can do outside of the typical physiological range that you could do with an antibody by using aptamers. Part of that robustness I already mentioned is temperature. But just going a little further, you don't need cold chain for these reagents. Storage, handling, shipping, you can do this at room temperature. They can stand a lot of heat. It really is remarkable how stable they are. And you can save a lot of money using this. So I remember when Rich Thayer and I were visiting a big diagnostic company many years ago talking about this. And we were talking about the fact that you don't require cold storage. And we had the head of manufacturing there. And he did a little calculation quickly, and he said, wow, we could save at least $100 million a year with no cold chain. So yeah, I think people tend to underestimate how important this could be. And of course, it's very important in things like looking at middle-income countries and point of care and situations where storage and environment are much more difficult to control. And also time to market. It's much faster to make these than it is to make most antibodies. And then also select them, determine what their performance is, and to manufacture them, because it's basically standard DNA synthesis. And that also leads to a lower cost of goods. It's not hybridomas in culture, it's standard DNA synthesis with some new chemistries. But people have tried to focus on things that can be done within the sort of phosphoramidite chemistry that most people are using today. And this can be done at a very high scale. So those are the attributes, but one of the reasons that you can get to these sort of attributes is the selection process. How it is that you can actually select the aptamer that you want. And so we'll talk much more about that here. Now, there are many company-specific names for aptamers. You'll see optimers, somomers, clickmers, Aegis bodies, HPAs, all kinds of stuff. There are differences, but most of the companies that are out there still use which are referred to as native DNA forms of aptamers, where it's just native nucleotides. But there's been a lot of work increasing the chemical diversity of aptamers. And I'm going to talk quite a bit about that here because I think it's extraordinarily important for what can be done with them. So let me talk a little bit about those sandwich assays again. And this time I'm going to try and compare a sandwich assay conducted with antibodies, which I've already shown. That's on the left-hand side of the panel. And then what I think we should anticipate for a properly designed aptamer assay. So again, we have the situation where we've got the two different wells, but now we're just going to look at positive samples. We're not going to bother looking at negative samples to complicate things, just positive. So in this case, we will add the sample to both of those wells, and now we're going to get binding again, and we get that specific and non-specific binding in the case of the antibody. But in the case of the aptamer, if we've done what we need to do, which I'll explain, there should be some binding which occurs to the surface, but most of the binding is going to be specific to the capture aptamer. And so now in the next step, where we're adding the label, if we've done that properly, we actually are going to get far fewer non-specific binding events with an aptamer than with an antibody. All right. So again, this can be realized in most immunoassay methods because it's not the signal that's the problem, it's the signal to the noise. Okay. Now, I'm going to talk about this briefly later, but I'm showing here an assay that has two aptamers. You can do it with just one. The most important one of those two components is actually almost always the capture reagent. So you can use an aptamer as a capture reagent and then use an antibody as the second reagent. And we have some experience with that. That worked just as well. And there's some reasons why you're going to want to be able to have that as an option that I'll get back to. Now let's talk specifically about aptamers. Firstly, what is an aptamer? Many of you are probably aware of this, but aptamers are short sequences of engineered RNA or DNA. More RNA stuff has been done more recently, but traditionally it's mostly been DNA. And that these bind a specific target molecule or a family of target molecules. Now, I've been discussing mostly proteins here, but small molecules, I'll come back to that a little bit. Other things which are not proteins that can be used as well. These aptamers exhibit a range of affinities from a KD in the picomolar range, which is rare, but people have gotten there, to micromolar range. I would say most of the things that I've seen are in the low nanomolar range. And with variable levels of off-target binding, and they're sometimes called chemical antibodies, which is a name I hate, but some people call them that. Now, this is a fairly long history. This goes back to a number of groups in the 90s. Firstly, Larry Gold and Craig Turek, they played a pivotal role in this whole area of discovery and development of aptamers. In 1990, they successfully isolated the first aptamers, which were RNA ligands capable of recognizing DNA polymerase of bacteriophage T4. They were inspired by biological evolution to create a revolutionary technique called SELEX. You probably have seen this. It stands for Systematic Evolution of Ligands by Exponential Enrichment. And most everybody in this field is using some form of SELEX. This method involves cycles of selection and amplification to identify aptamers with the characteristics that you want. I'm going to talk about that in a moment. Also then, simultaneously and independently, Andy Ellington and Jack Szostak, similarly to Larry and Craig, also developed a method for in vitro selection of RNAs. They're credited with coining the term aptamer, as a matter of fact, in a publication in Nature. And Jack's research on the evolution of RNAs and their role in the origins of life led him down this path. I mean, it really was because of those explorations he was doing. Andy's lab continues to work on developing functional nucleic acids for practical applications, including aptamer biosensors. And then there's Gerald Joyce at Scripps, and like Jack, he also conducted research on Darwinian evolution of RNA and DNA. He still does a great deal of work in that area. He also developed SELEX simultaneously with Larry and Craig and with Jack and Andy. And then I wanted to mention, and I'll come back to this, Steve Benner and his group had the idea of using extra artificial bases, first practicing what they called their Aegis system, which they're still doing a lot of work on, which artificially expanded genetic information system. So, and you'll see why it's called that in a few moments, but there's been a lot of clever things done with that. That was first reported about a decade ago. And since then, there's been just an amazing amount of innovation that's occurred over the last 15 years in this area. I'm going to tell you about several aspects of that. And if you're interested, there's a paper called Three Decades of Nucleic Acid Aptamer Technologies, which I found to be a very interesting read. It's a bit dated, but I think it does a pretty nice job. Given the history, I've got to say it's been a very long time in coming to get to high quality aptamers for immunoassays. Many of us in the diagnostics community worked with aptamers ever since the early 90s. We just could see the potential of these. For instance, when I was at Chiron, I funded a project with Andy Ellington and a few others in the field. The promise was so high, but the technology was just a bit early. We saw good binding events, but the selection processes hadn't been worked out quite as well. But I've kept looking to see when it was ready for the kinds of things I wanted to do. And finally, in 2007, while I was CEO of Tethys, which I've talked about in a number of different episodes here, which is a proteomics company in Emeryville, California, we had heard from some of the people we knew at SomaLogic that they had made some major advances. So my CSO at the time, Mike McKenna, and I went out to visit Larry Gold and his team in Boulder. Now, at Tethys, we had developed more than 100 different immunoassays on the Singulex platform, but we knew we could use better binders. And we were really focused on developing a lot of assays. We ended up developing almost 300 different assays for screening for biomarker panels. And we knew we needed better binders. We were constantly looking for things that we could use. I'll explain some of the improvements a little bit later here, but the bottom line is that we were very excited about what we saw from Larry's team. We made an agreement with them that we could obtain 7 of their aptamers that were going to be used in our case as capture reagents in immunoassays we already had, which were sandwich assays. And we had done that in the Singulex platform. So we obtained those aptamers. We then actually very quickly were able to just modify our assays so that we, instead of using a monoclonal for capture, we used the aptamer for capture, and we were blown away. I mean, all 7 assays were improved. The limit of quantitation, the dynamic range, the CVs. I mean, with very little optimization, they just were so much better. So I tried like crazy to get the guys at SomaLogic to provide us with their, now what they refer to as oligomers for our immunoassays, but they decided to go a different way, which that way was, of course, to develop the SomaScan technology with their aptamers, which is a great proteomics discovery platform, but I wish we'd been able to use those materials. But, that convinced me and all the people that worked with me that this approach was going to work for standard immunoassays. And I'll come back later to not only why they work so well, but the fact that I think we're suddenly seeing the applications that I was expecting. So I'm very disappointed we couldn't get access to the aptamers, but it was clear to us that this methodology does work. And so for all these years since then, I've still been very eager to find access to aptamers that could be used in immunoassays. And I'm going to tell you more about that in a few minutes. Let me explain a little bit about the modifications that Larry and team had made here. They had started off, like everybody else, with sort of your typical aptamers, which were just composed of the native 4 nucleotides that are found in DNA. And some very useful aptamers were found. But there are a lot of failures. What they decided to do is to actually create a greater diversity with these aptamers by adding different types of organic moieties to them that would change the binding and give them more diverse binding. So specifically, they were using uracil derivatives, and they added to it, for instance, a benzyl group, a naphthyl group, a tryptamino group, isobutyl group. So now we've got some very different sorts of things that are hanging off the end of those bits of DNA. We've known for many years that making modifications at that position within a uracil did permit enzymes to recognize these nucleotides. If you wanted to use that, you could use nucleases, you could use polymerases. I'm not sure how much was done with these derivatives there, but they had good reason to believe that making modifications at that point would give you good, stable hybridization, which it did. You know, I was reminded when I first saw this of what my friends in drug discovery used to say was the methyl ethyl futyl approach to drug design. So I was a bit skeptical at first, but in fact, this does work. And on the right hand side there is a table from the guys at SomaLogic where they were looking at specific target proteins, and you can see them listed on the far left, and they had used derivatives of the aptamers where they had a standard T moiety at that position. And you can see that in most cases, they failed to get a strong binder. But then they also tried their aptamer libraries containing the benzyl, the isobutyl, or the tryptamino. So, and you see that sometimes they failed, but sometimes they got nice binding events. And that's what they were hoping for, and that's what they saw. And I'll show you this has gone way beyond this now in many situations. But this was one of the major steps forward. Okay, so now what I'd like to do is generally describe the selects method and how it is that people select aptamers. So what's done here is first creating a library of aptamers. And those libraries may be, like I showed, the benzyl or the naphthol. There could be a library that has just those derivatives with the native bases. Sometimes people do combine these things, seeing that more and more. And then what you're trying to do is select from within that group. And often people have as many as 10 to the 10th different components within that particular library. And now trying to find the ones that have the binding properties that you want. So you start with the library. That's usually created by single strand DNA synthesis, standard way. You now have those materials together. You you have one means or another of actually combining the target molecules you're looking at. If I'm looking for a specific protein, I'm typically not going to use the complex blood sample to start with. I'm going to look at the protein itself that is of interest and try to find binders to that. But there'll be some other aspects of this that will improve my chances of success. So the first thing is I'm taking my library, I'm combining it with my target molecule. I'm permitting the aptamers to fold properly and bind to this thing. There are some tricks of the trade here I'll come back to about how long you let that incubate. Then you remove the unbound oligonucleotides from the library. There are a number of ways to do that, and I'll show you in a few moments a specific way people do it. But after that separation event, people typically use PCR to amplify what's left in the binding pool. So usually that means they've put primer binding sites at the end of their aptamer for all the components in the library so they can amplify those up and sequence them and determine which of the particular components has been binding. And also looking for whether there are commonalities in those binding sites. So we've now got those oligonucleotide products. We've analyzed them. We now have an enriched library for that. And we often go through a number of cycles of doing this to get better and better binding events. That is very typical. 5 to 10 cycles, not at all uncommon. There are different ways people do this, but that's the general idea. Start with a library, get the specific binding, move through this, and find the best binders you can. And then what is often done is a second round of this, where now instead of looking at the target molecule, I'm going to be looking at things that I'm afraid that it will bind to that I don't want it to bind to. Okay, so that is now a negative selection. That could be related proteins. You know, I've got something else that's going to be in that sample that's closely related, so I don't want it to bind to that. It could be that I've got a modified protein I'm looking for, like something that's phosphorylated, and I don't want the non-phosphorylated form. I can do that to negatively select. I can do things such as use a negative sample. I could actually use a blood sample that doesn't contain the antigen of interest and try to make sure that nothing in that sample binds. There are lots of things you can do here. You can actually look at the solid phase itself. In fact, the solid phase that's treated with, let's say, a gamish of things that you typically put on it because you know that can decrease the non-specific binding, but not perfectly. So you might be able to select that out. There are lots of options here. And that's one of the beauties of this is being able to do that. But one of the very specific things I wanted to mention is back to that incubation step. Here is where SomaLogic made another advancement. They had been using rather short incubation times. And when they did that, they mostly were seeing things that had a fast on rate and a fast off rate. It wasn't until they permitted things to sit for some time and let the fast off rate things come off that they got the best binders for a lot of these assays. You want things in most cases that bind quickly and then don't come off. Now, that binding quickly is also something you can deal with. For instance, in something like a lateral flow assay, you really do need to have very quick binding, just given the nature of how the assay works. Something like an ELISA, you can wait longer. You can have a longer incubation time. So you can actually select for those performance criteria in this process, which is just beautiful. Very difficult to do something like that with antibodies. It's more or less, a matter of um, uh, looking at a large number of different antibodies and hoping that you get the one that you want, whereas this is very systematic. You may not find what you want, but you have a very systematic way of determining whether it's there or not. There's some other very specific things you can do in this selection process, which I haven't seen many people do, but I just wanted to mention. For instance, you can use non-physiological conditions for some of these assays. We're kind of stuck with antibodies and using conditions which are very much like what you'd find in a human sample. But with an aptamer, you can heat it up. You can add materials to it that wouldn't normally be stable for antibodies and antigen complexes. In fact, there are situations where you want to break up antibody antigen complexes that already exist because there is a host antibody bound to an antigen. Very specifically, for instance, in HIV infections. After someone seroconverts, they now have antibodies against the virus. And so it's very difficult to perform an immunoassay unless what you do is degrade that antibody antigen complex, which people do with trick steps like with ADSs and things. But I think here you could just use conditions that destabilize that and just go ahead and perform the assay. In the example where you destabilize the antigen antibody complex in HIV, for instance, or also in HCV assays, you have to treat, you then have to quench that, you then have to add your reagents. It's multiple steps. This will be one step. You're done. So you can do a lot of things you couldn't otherwise do. All right. So the next slide here is just a specific example of a selects process. And unfortunately, this one goes counterclockwise instead of clockwise in the previous one. But it's the same idea where you're using the library, you're incubating it with the target, you're doing washes, and what a lot of people use is magnetic beads. Very convenient to do the assays on beads. Then you follow that up with the RT-PCR, and you look at the materials that have been amplified because they're the things that bound. And now you're looking at NGS usually as the output, and then you're repeating this. So those are the kind of things people do. So one of the issues with aptamers has been the percent of the time that you get an acceptable aptamer, and the number of reports that's on the order of 70% today. That is 70% of the time you get an adequate aptamer. But I've talked to a number of different experts in this area, and I do think we're going to see this rise from 70% up to 90 or more percent in the not too distant future. I think some people may already be there. And a lot of that has to do with the diversity, the chemical diversity again. Now, in the example I showed so far from SomaLogic, those are hydrophobic moieties. So no surprise, it usually binds to a hydrophobic portion of a protein. Um, so the accompanying problem with that is you often find that you can only get one aptamer per protein. That is something that I think probably led to SomaLogic doing what they did for SomaScan, which is using a configuration, very clever assay design where you only need one aptamer. But do recall that I mentioned that when we were at Tethys, we use only one aptamer too, because very convenient and very useful to have a capture antigen and then an antibody for the label. So that works beautifully, and I think may be adequate. But I think as we see more and more of this chemical diversity, you're going to see more and more um, uh, options where people are getting more than one aptamer per protein. And I know of a number of people who have already done that, but it's just not always the case. So beyond what I mentioned with SomaLogic, let me give you a few more examples of people doing interesting modifications. Um, so one is from the company Apis Assay Technologies. And in this particular slide off their website here, they have what they call clickmers. Clickmers are based on the Nobel Prize-winning click chemistry. If you're familiar with that, very clever chemistry. And they're using it to build unusual moieties, still using a single strand DNA oligonucleotide as a base here, but to increase the chemical diversity yet again. So they have used the native bases, but also modified bases as well. So uh, they haven't shown much about this, but I think they're being a little bit cagey here. But they have talked about the fact that they're looking at amino acid moieties, side chains, in what they're doing here. And I suspect that that's one of the reasons they're having a fair amount of success in developing aptamers that others have had difficulty with. So in the next slide here, also from their website, is so this step one, step two, where they're talking about this nucleotide derivative that they have, ethinyl uridine, in that same position because that works so well, 5 position. And so that then is a click chemistry approach where you use a specific type of molecule that will bind under the conditions where you have copper in the sample. So very clever, very, very efficient. They're they've got a partnership for GMP manufacturing of these in place, which is very good. It's very fast. They're able to make these materials rather quickly. It's a good group based in the UK. Again, this is from the Apis group. Something else from their website. On the left-hand side here, you see a situation where you've got a really interesting one because they're looking at flavy viruses. You've got tick-borne encephalitis virus. They've got yellow fever, West Nile, Japanese encephalitis, Zika, dengue. It's been difficult to get really good antibodies that don't cross-react with those other viruses. So they're showing a situation here where they're looking at all of those different viruses and then showing the specificity of the aptamers that they have created for 3 of those targets. So I think that is good evidence for the fact that you can, through the appropriate selection process, get to very desirable performance characteristics. Unfortunately, there's not a whole lot more that I found from them to say that this is generally applicable. They do say that they're also able to make aptamers against small molecules. That has been a problem in the past, but this has become something where more and more people are doing it. I'll show you another example in a few minutes. But early on, there was a lot of success looking at an aptamer that could differentiate between theophylline and caffeine, nice small molecules, just a very small shift of a methyl group there. And so everybody got very excited. Wow, we've got a new way to make these reagents against small molecules. And then years later, we had not seen very many examples where that worked. And now I think we're going to start seeing more of that again, which is very desirable. The next one is something I mentioned earlier, this Aegis approach. And this is from Firebird Biomolecular Technologies in Florida. This is really quite remarkable. And, I've got some appreciation of this because we worked with Steve Benner and his group many years ago when I was at Chiron looking at one of these unusual base pairs that we're using the bDNA assays. So it worked incredibly well. So the whole idea here is to literally create a new uh, genetic information system. And they've done that. And this has been used for a whole lot of things. But what this aegis approach is doing is giving you two new base pair types. They refer to them as Z and P, S and B. And I know that the Z and P were named after the people who worked on this, the husband and wife team, Zuni and his wife Pei, I think is her name. I don't know for sure, but I wouldn't be surprised if S and B is Stephen Benner, but I can't be sure. Anyhow, I know that they've been working on this for a long time, and it really does provide supplemental chemistries that fit perfectly in just standard DNA synthesis. So you can just use these in addition and expand that code and expand the binding capabilities. So they've gotten some very good results with creating these what they call aegis bodies. And I think this is another one of the major advancements in the use of aptamer chemistry and the expansion of the chemical diversity. And they've done some very interesting work shown here, actually even looking at binding to cells and using a selects process for that purpose. And again, starting out with the library. In this case, is a 6-letter code. They were using just one extra base pair instead of 2 extra base pairs. So, and that worked extremely well for finding aptamers and these aegis bodies that could bind specifically to specific cell types. So, and then they were able to actually use NGS the standard way, and they knew how to read for the ZP base pair, and it just gives them the results right away. I mean, it's just a very beautiful, simple, and elegant way to create aptamers. So here we are. We've got these additions to our chemical diversity. We've got, I just showed before with SomaScan, SomaLogic, the 4 types they've got, but this has now been expanded to a number of other side chains that can be employed in library design. I mentioned the Apis assay technologies, and I've just shown the amino acid side chains there. I don't know which ones they're using. And then with SomaLogic, the example of the expanded base pairs that can be used in parallel. So there are other people that are doing similar things. One is Velocity Sciences, which was started by a number of ex-SomaLogic people. It's also in Boulder. They're using RNA backbones instead of DNA backbones. I mean, we've got a lot of things going on here, but we now have quite a diverse group of options. And I'd like just talk a little bit about some of these companies that are out there and some of the options that are available to any of you that want to get into this area. So I did mention before that I was going to focus on the non-native aptamer companies, but there are several of those companies out there that have been successful in this area. There are companies such as BasePair in Texas that has been active for a very long time and done a lot of very good work. Aptitude out of Santa Barbara, Aptamer Group, Pentabind, Aptogen. I have not had direct experience with any of those 3, but they are well regarded. Some of these companies actually have libraries of aptamers that you can get access to. And the one I'm showing here is from the Aptamer Group. They have a number of them to both small molecules and to proteins that you can get access to. I don't know whether the characteristics of these are going to be such that they are going to work in assays that are of use, but I think that's an interesting thing to provide. Other companies are doing this now as well. And then, but the expanded chemistry companies that are willing to engage and conduct R&D and provide reagents to you for the libraries are Apis Assay Technologies, I mentioned, and also Firebird. And I think those are both excellent companies I would definitely work with. I have tried for years, as I said, to work with SomaLogic. Now they've merged with Standard Tools. Back then, as I mentioned, they went down the path with SomaScan. I never could convince them to use any of those materials to let us do the immunoassays and commercial assays. I haven't even tried it in a long time. I don't know, but I wish they would, because they've got such a wonderful library of 11,000 different aptamers that they're using that could be used. I don't know if the performance will be appropriate, but I sure would like to try. The ones that we got, as I mentioned, they worked. So very nicely without modification. Now, at this point, what I'd like to have done was to show you a whole bunch of commercial and research immunoassays based on well-designed and well-selected aptamers. But I can't because they're still few and far between. Um, I still think there's a lot of skepticism about aptamers because they just didn't work early on. But my Tethys team and I had a great opportunity to test them, worked them out. So we had a relatively unique and I suspect biased view. You just haven't been able to do that. I don't know anybody else who got them from SomaLogic to do that. And they certainly wouldn't have let us do that 5 years later. So, but we came away recognizing that this works. This really does work. Now, recently, Altaris had the opportunity to introduce an academic group that had a very interesting high-profile protein molecule that had a post-translational modification that they wanted to be able to specifically detect in an assay. So we put them in contact with a company, an aptamer company, that we thought could create and select the appropriate sort of aptamer for them. And the person that developed that relationship was our colleague, Sarah Hamren, who was the person at Tethys who was responsible for our aptamer studies with SomaLogic. So, she was as biased as I am, but, we found that that company was able to make an aptamer for that molecule, and it works very well, and it's been incorporated into an immunoassay that works extraordinarily well. That is something that we expected to happen. We just knew it would work because we'd seen it before. So I just don't think that the advantages of aptamers are widely appreciated and realized, and therefore not used. The performance, the robustness, the cold chain, the cost of goods, unique ability to select the properties that are needed. I mean, I just think it's underappreciated. So I would have to end with telling you, I think the time has come to bypass those ancient antibodies and join us in the future with aptamers. Aptamers are the way to go. You might end up using an aptamer and an antibody, but that aptamer will make all the difference when you replace it as a capture material for your assays. So thank you very much for listening to this episode of This Is Only a Test. This is Mickey Urdea for Altaris Associates. Thank you very much and bye.